here with Oliver Bonham-Carter. What's the name of your paper?
It's called, it's the BeagleTM, a text mining approach to finding different types of the actual title is, but it's BeagleTM, that's what I call it.
BeagleTM. Tell me about BeagleTM.
So BeagleTM is, imagine for instance that you are working in a research area where you're looking at different types of genes here, and you call your gene X, and I'm working on a similar type of research here, but I call my gene Y. And one of the problems is though that, for instance, I may not know that you're working on these genes at all. I may not know that you have a project in this area.
There's no standard for it.
There's no standard, and in my area in bioinformatics, one of the big problems is though that you have these different explorers basically who are going off and finding different types of genes, and they don't know, for instance, that anybody else is working on these genes, and so they come up with their own names on these things. And while that's fine, it kind of makes those genes become, or those papers become kind of isolated though, because unless I know that those genes are there, and unless I know those names, then I can't find them on my literature reviews. I can't find them in a search algorithm. And so what my BeagleTM paper does is it will go through and it will figure out, for instance, I'm looking out at not just genes, but I'm looking at, for instance, post-translational modifications, which are it's in that central dogma of biology here, where you have your proteins which are shaped into something, when after they're created, they're shaped. That's what we call the post-translational modification. Mm-hmm. But what that does is, where I'm looking for instance, genes which have a certain type of post-translational modification, and they're also maybe involved in some kind of an ailment, maybe some kind of like Alzheimer's or something else, and they're also involved with some kind of a stress. And so this is a machine basically it's a statistical machine learning method here that basically goes through and it will find the papers which are somehow that are connected by maybe 3 of those 4. So I don't know the genes, but I know that there's Alzheimer's involved, I know there's a post-translational modification involved, and I know that there's some kind of stress involved. And so this will find all of those papers here, and then it might find your paper. It's very likely to find your paper because you're using these same terms.
It should be about the same genes.
It should be about the same genes.
Yeah, of course.
And so what we do is we create what's called these relationship networks, and relationship networks are basically giant plans, I guess, where you have a node which represents a post-translational modification. You have another node which represents the gene, another node that represents the ailment, another one that represents the stress. And so the idea is we're making these kinds of what they call cliques in graph theory here, and you can just kind of follow these cliques and say, well, here's one of my players, here's another one of my players, here's another one of my players, and this is the final unknown actor. And so then I can find these papers just by knowing 3 out of the 4 possible nodes here.
And that there should be a match.
There should be a match. Yeah. But another nice thing about this paper, though, is that if there isn't anybody working on these genes, supposing there's no literature, for instance, that's working on, for instance, a gene which would somehow connect Alzheimer's with acetylation, which is a post-translational modification, and some kind of stress. Then, for instance, I might be able to look at this click and say, there's one missing. Maybe there's a connection. Maybe all of these players here, there should be some gene out there that has something to do with these other 3 players here. Maybe there's some gene out there which still has, which exists somewhere. And so this is actually kind of hinting at maybe potential projects that you can use, because you can see if you're looking for 4 main actors here. If you have 3 of them which aren't there, or sorry, if 3 of them are there and one of them is missing, then you might be able to think, well, perhaps there is.
It hints at something more.
It hints at something more, exactly. Yeah. And I think that when you look at some of the new studies that have come out in my work here, when you look at some of the studies, like the new cutting-edge studies that have come out, we can almost predict that these are coming out there. We knew that there was a gene that had something to do with these other 3 actors here, but we just didn't know what the name was. And so you just keep running this thing after each session here, after all the papers come out, and then eventually you'll find, oh, there is a gene. Someone has found this gene that connects these other ideas together.
Yeah. Okay. And is BeagleTM being used at this time?
It's being used in our lab. Basically, it was developed originally, though, because my colleagues and I, we were working with post-translational modifications, and one of the things is that when these post-translational modifications are actually quite important because we're looking to see how certain proteins are modified after they've been created. They're modified to deal with certain types of stresses. And one of the problems that we ran into was that there are many papers out there which may mention a post-translational modification, and it may mention also the gene or the protein or the ailment, but those were not terms that were in the keyword bank. Yeah. So for instance, we had to know those papers, and we had to actually read those papers and know that there was a connection there.
So you came up with it because you had the need to
find those papers. We needed to do an extensive literature review, and so we had this basically an algorithmic approach to find some of these papers, and it's actually helped us tremendously in our research, though, because we're looking for a number of papers for which the authors themselves never specified that this was a paper about, say, acetylation, or maybe they never specified that this was a paper about a specific type of ailment. Yeah. In fact, those terms were just thrown into the paper as a kind of an afterthought, and we never. It was as if the author wasn't very creative, perhaps, when they put the keywords together, or maybe the keywords were too vague, or something like that. Yeah. And so we're actually finding, we're using more than just metadata to find out these papers. And so I'm hoping that this is like a new way of looking for literature review.
And will BeagleTM be available for other researchers as well then?
Yes, it will. So we are planning to put it out onto GitHub. And so the idea is that, like I was saying, my area is actually bioinformatics. And one of the big problems in bioinformatics is that you have these. Bioinformatics is the child of computer science, mathematics, and biology. And so when you have researchers from all these different groups coming together, they tend to use different languages to name the same thing, which is actually quite a big problem. And so, for instance, using something like BeagleTM, it will help them to perhaps find some of these papers where the vocabulary may differ or vary from paper to paper. One thing that comes to mind immediately now is in computer science, the word MAC might be the MAC address, which is like an IP or network type of word. Whereas, for instance, MAC is also a name of a protein, and in mathematics, MAC could be like the math, some kind of a math attendance center here.
There's too much overlapping.
There are too many. I mean, everyone has an acronym that has MAC. But so the idea is that, for instance, if you combine the word MAC with maybe other types of terms that would be reasonable for your research, then it will be able to kind of take that information and combine it with other information, and then you'll be able to find potentially your paper that you're looking for.
Yeah, that's awesome.
It's very exciting. I think that it's very exciting because one of the things is that it goes through the PubMed papers, and so it reads something like 16 million papers in seemingly an afternoon. And so when people ask me, when they ask me like what my day was like, I'll say, well, today I read 16 million papers.
That's a good day, right?
Yeah. It's not bad. It's not bad.
Okay. Oliver, thank you so much.
Thank you very much for your time.
You've been here at the conference for 2 days.
That's right.
What was your highlight of the conference at this time? I
think that it's not just one. It's been many different highlights here. One of my areas is machine learning, and there's a machine learning track here, which I think is actually quite exciting. I've had some very interesting conversations with people who are using machine learning for some very interesting ideas that I'd like to put into bioinformatics. I think that machine learning has like really no limits to what it can do. Yeah. And so I think that my area, though, I'm looking through like hundreds of millions of different types of data, and so having a machine help me with that, in protein, when I'm looking at protein data here, for instance, having a machine help me with that would be actually very handy.
Yeah, of course, especially in your field.
That's exactly.
Okay. What was your favorite keynote thus far?
I would say that there was a person who was speaking yesterday from, was it Microsoft? I think it was about. They're talking about yes, that's right. I thought that was very, very interesting. It makes me. I enjoy these types of keynotes because I think that it helps us to maybe dream bigger. Perhaps I think it maybe helps us to come up with more ideas on, for instance, on what we're doing. It helps us to put everything into perspective. One problem I do find though is that when you're working on the same types of research projects all the time here, you tend to develop some kind of tunnel vision, and so it takes somebody who has these broad, far-reaching ideas to help you break out of that tunnel vision and start seeing maybe a bigger picture, maybe a more global view of things. And so I think that these keynotes, and all of them, are very important.
So this conference helps you with widening your view?
Oh, certainly. I think it's very important. And I've made some good contacts too. I think I've had I've made some people who may be potential collaborators in the future.
That's awesome.
Yeah, so I think it's been
Oliver,
rewarding.
thank you so much.
Thank you.